Okay, Dr. Mitton here. I'm going to give you a quick screen recorded movie of how to use the University of Santa Cruz genome browser. The first thing you'll want to do is go to genome.ucsc.edu. When you land there, it should look like this. And the reason we're going here is to get images like these, which are gene structures in the genome, showing intron and exons. You'll also see the chromosome location. You'll want to do this with the gene that we will discover causes Pingolap version of chromatopsia. I'm just going to show you how to do it with some other genes. You'll be able to do the same thing. Normally, you're probably defaulted. If you just search right away, it'll probably just default and you'll be doing the human genome. And let's say you type in Rho. Rho is the official acronym for the rhodopsin gene. Sometimes I'll get a little list back here. It's often usually the top one, but there's rhodopsin. That's the human rhodopsin gene. And you can see you might get here, and there's a lot of different tracks here with different views. I can show other ones here. Let me just flip on a couple of things. You might get here and find that you have, depending on how your browser is set, let's do a refresh on this. You might come here and maybe it's a little bit more complex with more tracks on here. No matter what's on, all I recommend that you do is you leave the base position to be dense. You can leave main on, and I would turn off all the other tracks that might be on anywhere in these lists. You can hit any of these refresh buttons at any time, and it just redraws the view. So here's rhodopsin. It's come up. Thick bars are the exons. The thicker parts of the thick bars are coding regions, and these thinner parts of the thick regions are non-coding. And in between are the thin lines that are exon sequence, where this gene is relative in the chromosome is shown in the chromosome ideogram up here. This is for chromosome 3. We're in a band region, which is traditionally called from traditional staining methods. It's in the Q arm, and it's 22.1. Whether you're in the Q arm of a chromosome or the P arm of a chromosome, the Q or P values start low near the centromere, and they increase as you go out to the ends of the arm. So here on the chromosome, you see where you are physically on chromosome 3, and the region being shown to you is usually up here, chromosome 3. You can see this is the base number in the chromosome that are being shown here in this window. The little chevrons show you the direction of the gene. We're kind of looking, if you're thinking that you're looking at this backwards, what this is telling you right now is looking at this chromosome. This would be on the bottom strand. Going that way, you can hit reverse if that happens to be on and flip any gene around to look at it. And you can see the chromosome ideograms flipped around, and so is the gene structure. If the labeling doesn't look great to you, you can play around with things. There's a configure button right under the main image. I usually recommend that if this isn't set wide to 12 or 1600 pixels, make sure you set it fairly wide. Text size, if you're defaulting at a lower text size, I would say select 24. Anytime, and then you just resubmit. Hit the submit button. It redraws the view for you. What you'll want to have is when you get the gene of interest, Pingolap, that causes Pingolapese chromatopsia, one thing you have to do in your term paper on that subject is to have a figure that shows us the structure of the gene, the relevant gene, and its location. and to make a figure on that. And to do that, you will get these two images down in the forms of PDF images. They insert very nicely into Word documents, and they're pretty easy to work with. And PDF documents also, as you scale them to any size, they look good. They do not get blocky. And that's because they're written with a PostScript mathematical type of graphical language. You will go to View menu when you have the view the way you want it, PDF. And there's two options here. Downloading the current browser graphic in PDF gives you that chromosome exon intron structure diagram. The other link gives you the diagram of the chromosome with the bands and where you are. So all you do is download both of them. Your browser might view it and then you'll download it. Make sure you get a download of the file. I'm going to do that. And I'm also going to get the ideogram of the chromosome, as you see here. Let me download that. So they downloaded into my space. And if I bring them up, I'm just going to drag mine up into the view window here. Here's what they look like. So that gives us those files. And that's how you will find your gene of interest.